Now then, tonight, we're going to be talking about cavefish. There is no eye on that fish. There is zero eye on that fish.
My name's Thomas Merritt. I am a professor at Laurentian University. I wonder what's been going on in the world of blind cavefish. And actually, a lot.
SciAnts. We get experimental science. We're curious, non-judgmental.
Now then, tonight, we're going to be talking about cavefish. Now, cavefish mutations lead them to have, within a same species, there are blind and not blind cavefish. The ones that live in the cave have no eyes. The one that lives outside has eyes. Now you might be, Balint, that's very goofy. How does something like that happen? The cavefish that have no eyes actually form eyes during development. Like as they're undergoing development, they form totally normal, fully functional eyes. And it's like, well, that's weird, right? Like, why are they're forming fully functional eyes? So what's going on there? On top of forming the fully functional eyes, then those eyes undergo cell death and they just all die. All that tissue gone. Or a blind. So if you move Ninja, the blind cavefish, the regular water, those cells still die because they've picked up a mutation during eye development. And today we're going to have on Professor Thomas Merritt to come join us. And we're going to be talking about cavefish. That there is no eye on that fish. There is zero eye on that fish.
My name's Thomas Merritt. I am a professor at Laurentian University in Northern Ontario. That's about 4 hours north of Toronto. And you and I know each other from my work in fruit flies, but I do work in various other systems, including a very long time ago, a brief stay in the world of blind fish.
This is the Munchkin, Thomas. So she is excited to be here. She just got home. Yeah, that was you. We're talking about some fish today. Yeah.
So as a PhD student, I worked on gene evolution. And we're actually working on a set of genes that are expressed in the eye and brain of fish. And they have these really interesting adaptations to being in that environment. And somebody that I was working with, it was probably my major professor, was like, it'd be really interesting to look at these genes in fish that don't have eyes and see what happens. But when you were talking about, we should do some spooky science, I was like, I wonder what's been going on in the world of blind cavefish. And actually a lot. And one of the really cool things about the fish is that there are a surprising number of genes that seem to be completely unaffected by the fact there are no eyes. And so why is that a head scratcher? It's basically the idea is here's an eye gene and here's an animal with no eyes and the gene looks absolutely normal because it actually is absolutely normal. And one of the sort of punchlines of that is this, what we've learned in the last 30 years is that genes almost never have a single function. And so in this fish, the function is a little bit different. There's not the eye role, there are other roles. And so they're just basically there's this whole suite from genes that seem to be really involved with the evolution of this blindness in fish, and then a set of genes that have absolutely nothing to do with it whatsoever. And they're just cruising along happily with other sorts of functions.
We were actually looking at one of the papers that you sent us beforehand that was showing that everything like during the embryo stage to kick off is totally normal, that they're developing along normal eyes, and then all of a sudden they just start undergoing cell death with the eye tissue, which is kind of wild to think about, that you're investing that much energy to begin with in the development of the eye and then only to go kill it. Or are we thinking of it wrong?
No, well, okay, yes, actually you are thinking of it wrong. But so the really hard thing to wrap your head around with these systems is things are not evolving to anything. So the fish is not evolving to a more efficient eyeless state, but we're not evolving to being eyeless. We're evolving from having eyes. Like, okay, well, why is that bad, right? And so maybe that's the interesting way to think about this question is, why would you lose eyes in the first place? And, you could think, okay, well, they're just accumulating errors because there's no pressure to have the eyes. But it seems to be happening repeatedly. And the Tetris situation is actually really cool because you've got these central populations that are completely sighted, and then you've got multiple, apparently independent populations that have lost sight. There are a couple of things that are super cool in this. One, it looks like different populations have turned off eye development at different stages, which, if you buy my argument that we're not evolving to anything, makes perfect sense. So if that's true, then it's likely that in different locations, different genes might be turned off because it's not somebody going in and saying, okay, what's the most efficient way to do this? We're going to efficiently do it here. It's basically somebody with a shotgun taking a pop shot at the genome. And if you happen to hit a gene that knocks out eye development, then that's a reproductively successful strategy. And so you're going to have different strategies in different populations. One of the questions might be, what else is changing? And if you think about the environment that it's in, what else is different between a cave and an open pond? All of the caves are a restricted, like physically restricted environment. And so these individual populations of tetras have lost eyes. We talked about the hybridization. You can hybridize the cave tetras. And when you do, they don't always have no eyes. And what that's telling you is there's an independent genetic mechanism between those two populations. And this is super important. If you go back to this idea, we don't evolve to something. The fish is not evolving to some sort of optimized state. It's just evolving from sighted to not sighted. And what path it takes, it's a series of random things. And you can imagine, okay, there's one gene that has to be turned off, and then you don't have an eye, and that's more efficient. It's just, biology doesn't work that way. And there's almost never one magic switch. There are 15 switches that all come together to contribute to these eyes. And if there are 15 genes and you've got 6 different populations, then they're likely to land on different genes in different populations.
And that's exactly what you were saying too, of like the different isolated populations, they're not all identical in terms of what's changed. So it makes total sense then that if you do interbreed them, it's not just eye or no eye. It's probably a mix, right, of what ends up happening.
Yep. 100%. What makes an eye isn't the genes in the genome, it's what genes are active, what genes are being turned on or turned off. And what this lab showed was that it's actually methylation of the DNA that is turning on or off the gene. And so there are a series of genes that are hypermethylated that are turned off, and you don't get eye function. So if you buy this idea of evolving from, then in some cases, the genes are actually non-functional. But in at least one case, the genes are functional because they're functioning in other places. Here, they're not being expressed because that region of the gene that's responsible for that regulation is hypermethylated and it's turned off.
One of the papers that you sent over that we were kind of marveling at was when they identified one of the genetic elements that were different in the cave fish versus the non-cave living fish. They were actually able to go in and perform this mutation and make a transgenic fish. So again, a fish for the chat that we've genetically modified by just taking out that gene, and now it's a blind fish.
Right.
So with that, there's a follow-up question actually is, we've been able to have this, like we can make the blind by just changing that genetic element. Are you aware of anything in the literature where we've been able to do the opposite? Like, is there anything like from the eye or the muscle or color where we can actually put back the function, or is that level more complicated than this one-to-one that we're kind of doing in the knockout?
Yeah, that's an interesting question. So it's much easier. It's like, every 4-year-old knows it's much easier to break things than it is to fix them, right? And it ends up that biologically, this is much like being a kid playing in a toy box where like you smash something like, oh yeah, that's broken. You can't just like push it back together and hope that it works. So there's another example. So there's a gene called PAX6 that's expressed in essentially everything. And we didn't know that initially. And one of the places that it was studied early on was in fruit flies. And the gene was studied in fruit flies because a mutation of the gene essentially eliminates eye development in flies. And so because we're fly biologists, it was named eyeless because we're much more creative than the mouse biologists who named the same gene PAX6. Like seriously, you had eyeless and you went with PAX6. If you express eyeless in somewhere other than the eye in a fly, you get an eye, but it's not a functional eye. And so you can express it in like the arms, the antenna, and you get these eye shapes, but they're not innervated. And so they look like a multifaceted fly eye. So that's an interesting case of this master regulatory switch. And that, whether it's PAX6 or eyeless, will actually function in the other organisms. So you can drive eye development with PAX6 in a fly, and you can drive eye development in a mouse with eyeless, but it's not a functional eye. And it ends up that it's a regulatory switch that's necessary but not sufficient to give you eye function. So I think it's likely the same kind of thing in these tetras where you can knock out the eye, knock out a gene, knock down a gene, There are lots of different permutations, but we don't have a case where it's come back. And part of it, it's really hard to fix something. So even just putting the functional gene back, there are lots of subtle changes in the genome that have to be there to get proper function. So even if you could repair the gene, it's very difficult to know how to repair all the associated regulatory elements that make that work correctly. Unless, did I miss any questions? Are we good? We sort of wrapped up.
I think we wrapped it up. Yeah. Thank you so very much. That was a blast. And yeah. Well, next time, y'all, we'll do some fungal parasitism. Thank you so much for your time and your insights as always. Have a lovely rest of your evening.
Thank you. This was a lot of fun as always. I appreciate it. Take care.
Bye, Thomas. Thank you. Guys, I hope y'all enjoyed that. I had a lot of fun. That was a good time. It was just cool to talk about like the evolution of the loss of the eyes in this fish and how that came to be and what that means. Like, there was a lot of cool science there. I hope you all enjoyed it as much as I did.